I am your host Deepak Verma, come with a new topic today, that is cellular energetics, and actually I am emphasized on energy transduction in mitochondria. The Before going to start, I am giving you a little introduction. As we know that the most important molecule for capturing and transferring free energy in biological system is adenosine triphosphate or ATP. Cells use the energy released during hydrolysis of the terminal high energy phosphoanhydride bond in ATP to power many energetically unfavorable processes. Examples include the synthesis of proteins from amino acids and of nucleic acid from nucleotide, transport of molecules against a concentration gradient by ATP powered pump, concentration of muscles and movement of cilia. Although other high energy molecules occurs in the cell, ATP is universal currency of chemical energy. It is found in all type of organism and must have occurred in the earliest life forms. This video or this video focused how cells generate the high energy phosphoanhydride bond of ATP from ADP and inorganic phosphate. So now we move to the aerobic oxidation. So how this proton motive force comes into this matter, cellular energetics. So actually in aerobic oxidation, fatty acids and sugar, principally glucose, are metabolized to carbon dioxide and water, and released energy is converted to the chemical energy of phosphoanhydride bond in ATP. In animal cells, the most other non-photosynthetic cells, ATP is generated mainly by this process. The initial step is the oxidation of glucose called glycolysis, occurs in the cytosol in both eukaryotes and prokaryotes and do not require energy. The final step, which requires oxygen, generates most of the ATP in eukaryotes. These later stages of aerobic oxidation occurs in the mitochondria. In prokaryotes, which contains only a plasma membrane and lack internal organelles, many of the final steps occurs on the plasma membrane. The final stage of fatty acid metabolism sometimes occurs in mitochondria and generate ATP in most eukaryotic cells. However, fatty acids are metabolized to CO2 and water in peroxisome without production of ATP. So there are two means of to produce energy. One is the photosynthesis, and this photosynthesis only is the plant kingdom, and one is the mitochondria. So now becomes actually chemoosmosis. What is this chemoosmosis? Chemoosmosis can occur only in sealed membrane limited compartment that are impermeable to protons. The proton motive force is generated by the stepwise movement of electrons from higher to lower energy state via membrane-bound electron carriers in mitochondria and non-photosynthetic bacteria. Electrons from NADH are transferred to O2, the ultimate electron acceptor, and in the thylakoid membrane of chloroplast, energy absorbed from the light stripes electrons from water and power their movement to other electron carriers, particularly NADP+. Eventually, these electrons are donated to CO2 to synthesize carbohydrate. All these systems, however, contain some similar carriers that couples electron transport to the pumping of protons across membrane always from the cytosolic phase to the exoplasmic phase of membrane, thereby generating a proton motive force, which is shown in here. Invariably, the cytosolic phase has a negative electron potential related to the exoplasmic phases. So this is actually overview of generation, and this picture depicts the overview of generation and utilization of proton motive force as a transmembrane proton concentration gradient and a voltage gradient, collectively called the proton motive force, are generated during photosynthesis and or aerobic oxidation of carbon compounds in mitochondria and aerobic bacteria. In chemoosmotic coupling, a proton motive force powers an energy requiring process such as ATP synthesis. Here I am showing what is A, what is B, and what is C. See? A and here I am showing you membrane orientation and direction of proton movement during chemoosmotically coupled ATP synthesis in bacteria, mitochondria, and chloroplast. The membrane surface facing a shaded area is cytosolic phase. Here, the surface facing an unshaded area is an exoplasmic phase. Note that the cytosolic phase of the bacterial plasma membrane, the matrix phase of the inner mitochondrial membrane, and the stromal phase of the thylakoid membrane all are equivalent. During electron transport, protons are always pumped from the cytosolic phase to exoplasmic phase, creating a proton concentration gradient. Exoplasmic phase greater than the cytosolic phase, and the electrical potential negative cytosolic phase and positive exoplasmic phase across the membrane. During the coupled synthesis of ATP, protons flow in the reverse direction through ATP synthase F0 and F1 complex, which protrudes from the cytosolic phase in all cases. Moreover, mitochondria, chloroplast, and bacteria utilize essentially the same kind of membrane protein, this F0 and F1 complex, to synthesize ATP. The F0 F1 complex, now commonly called ATP synthase, is a member of F class of ATP powered proton pump. In all cases, ATP synthase is positioned with a globular F1 domain with catalyzed ATP synthesis on the cytosolic phase of the membrane. So ATP is always found on the cytosolic phase of the membrane. Protons always flow through the ATP synthase from the exoplasmic to the cytosolic phase of the membrane, driven by a combination of proton concentration gradient and the membrane electrical potential. So I am not going to go in details with the glycolysis, just directly to the oxidation of glucose and fatty acids to CO2. The complete aerobic oxidation of each molecule of glucose yields 6 molecules of CO2 and is coupled to the synthesis of as many as 30 molecules of ATP. Glycolysis in the initial stage of glucose metabolism takes place in the cytosol and does not involve molecular oxygen. It produces a small amount of ATP and the 3-carbon compound pyruvate. In aerobic cells, pyruvate formed in glycolysis is transported into the mitochondria, where it is oxidized by O2 to CO2 via chemoosmotic coupling. The oxidation of pyruvate into the mitochondria generates the bulk of ATP produced during the conversion of glucose to CO2. Here I am going to discuss the biochemical pathway that oxidize glucose and fatty acid to CO2 and H2O. The fate of released electrons is described in this equation. There C6H12O6 plus 6O2 plus 30 Pi plus 30 ADP plus 30 protons gives to 6 CO2 plus 30 ATP plus 36 water molecules. As a set of 10 water soluble cytosolic enzyme, actually glycolysis is a 10 water soluble cytosolic enzyme catalyze the reaction continues to the glycolytic pathway, in which one molecule of glucose is converted to the two molecules of pyruvate. All the metabolic intermediate between glucose and pyruvate are water soluble phosphorylated compound. I'm giving you here the quick introduction. So in anaerobic metabolism, what happens into the anaerobic metabolism? I mean, eukaryotes are, as many eukaryotes are actually obligate aerobes. They grow only in the presence of oxygen and metabolize glucose completely to CO2 with the concomitant production of a large amount of ATP. Most eukaryotes, however, can generate some ATP by anaerobic metabolism. Few eukaryotes are facultative anaerobes. They grow in either the presence of or absence of oxygen, such as annelids, mollusks, and some yeast. They live and grow for days without oxygen. So, in anaerobes, each glucose molecule gives only the two ATP molecules. And acetyl coenzyme derived from pyruvate is oxidized to yield CO2 and reduce coenzyme in mitochondria. In reality, immediately after pyruvate transported from the cytosol across the mitochondrial membrane to the matrix, it reacts with coenzyme A, forming CO2 and intermediate acetyl coenzyme A, which is shown here. This reaction, catalyzed by pyruvate dehydrogenase, is highly exergonic and essentially irreversible reaction. I want to going to discuss that here. During the pyruvate dehydrogenase reaction, NAD plus is reduced, forming NADH. In contrast, during the reaction catalyzed by lactate dehydrogenase and alcohol dehydrogenase, the NADH is oxidized, forming the NAD plus. So this substrate level phosphorylation generates ATP during this. All the things are related to the glycolysis. So now we comes to our main topic, mitochondria. So mitochondria are among the large organelles in the cell, each one being about a size of E. coli bacterium. Most eukaryotic cells contain many mitochondria, which collectively can occupy as much as 25% of the volume of the cytoplasm. They are large enough to see under a light microscope, but the detail of their structure can be viewed only with the electron microscope. The outer membrane defines the smooth outer perimeter of the mitochondria. In contrast, the inner membrane has numerous invagination called cristae. These membranes define two sub-mitochondrial compartments. The inner membrane is space between the outer membrane and the inner membrane with its cristae and the matrix, the central compartment. The fractionation and purification of these membranes and compartments have made it possible to determine their protein and phospholipid composition and localized each enzyme catalyzed reaction to a specific membrane or space. So here I'm giving you this picture. This actually shows the internal structure of a mitochondria. This is actually a schematic diagram showing the principal membranes and the compartment. The cristae form sheet and tubes of invagination of the inner membrane and connect to the inner membrane through the relatively small uniform tubular structure called crystal junctions. The inner membrane space appears continuous with the lumen of each crista. The F0 F1 complex stored in red spheres which catalyze or I can say synthesize ATP. The inner membrane particles that protrude from the cristae and inner membrane into the matrix. The matrix contains the mitochondrial DNA, here it is shown in the blue strands, ribosomes, small blue spheres, and granules, large yellow spheres. And I think I should stop to here regarding mitochondria, otherwise I have to discuss outer membrane, then inner membrane, then matrix, all the things I have to discuss. Then, so actually I am going you here the cellular energetics, not this. So now I'm going to the direct to the summary of aerobic oxidation of pyruvate and fatty acid in the mitochondria. That is actually the function of mitochondria. So this picture is showing the outer membrane is freely permeable to all metabolites, but specific transport proteins, there's colored ovals in the inner membrane are required to the import pyruvate. This is shown in yellow. ADP is green and inorganic phosphate is purple into the matrix, and to export ATP green in color. NADH generated in the cytosol is not transported directly into the matrix because the inner membrane is impermeable to NAD plus and NADH. Instead, a shuttle system red here transport electrons from cytosolic NADH to NAD plus into the matrix. Oxygen diffuse into the matrix and CO2 diffuse out easily. So there is some stages. So stage 1, there are 3 stages. So stage 1, fatty acid groups are transferred from fatty acyl coenzyme A and transported across the inner membrane via a special carrier as shown in blue oval and reattached to coenzyme A on the matrix side. Pyruvate is converted to acetyl coenzyme A with the formation of NADH, and fatty acid attached to the coenzyme A are also converted to acetyl coenzyme A with formation of NADH and FADH. Oxidation of acetyl coenzyme A in the citric acid cycle generates NADH and FADH2. Now we come to the stage 2. So here electrons from these reduced coenzymes are transferred via electron-transport complex. Blue boxes, I am showing it here. 4 blue boxes to oxygen concomitant with the transport of protons ion from the matrix to the inner membrane space, generating the proton motive force. Electrons from NADH flows directly from complex 1 to complex 3, bypassing complex 2. And the last third stage is ATP synthesis. This F0 F1 complex is showing in orange color harness the proton motive force to synthesize ATP. Blue arrows indicate electrons flow, red arrow transmembrane movement of protons, and green arrow transport of the metabolites. So I think this picture showing a lot of things here. The whole function of the mitochondria. Up to here, we understand that most of the free energy released during the oxidation of glucose to CO2 is retained in the reduced coenzymes NADH and FADH2 generated during glycolysis and the citric acid cycle. During respiration, electrons are released from NADH and FADH2 and eventually are transferred to O2 forming H2O according to this equation here. NADH plus proton plus half O2 is changed to the NAD plus and H2O, and similarly FADH2 plus half oxygen is changed to the FAD plus H2O. So the delta G, the free energy change values for these strongly exergonic reactions are highly in minus kilocalorie. For NADH is about 52.6 kilocalorie per mole, and for FADH2 it's minus 43.4 kilocalorie per mole. So recall that the conversion of one glucose molecule to CO2 via the glycolytic pathway and citric acid cycles yield that 10 NADH and 2 FADH2 molecules. So oxidation of these reduced coenzymes has a total delta G of minus 613 kilocalorie per mole. By this calculation, simple calculation, 10 into minus 52.6 plus and 2 plus minus 43.4, this comes to around 613. Thus, of the potential free energy present in the chemical bond of glucose is 680 kilocalorie per mole. So, about 90% is converted. 90% is conserved in the reduced coenzyme. The free energy released during oxidation of a single NADH and or FADH molecule by O2 is sufficient to drive the synthesis of several molecules of ATP from ADP and inorganic phosphate. A reaction with the delta G of plus 7.3 calories per mole. The mitochondria maximize the production of ATP by transferring electrons from NADH and FADH2 through a series of electrons carriers, all but one of which are integral components of inner membrane. This step-by-step transfer of electrons via respiratory electron-transport chain allows the free energy NADH and FADH2 to be released in small increments and stored as a proton motive force. So at several sites during electron transport from NADH to O2, protons from mitochondrial matrix are pumped across the inner mitochondrial membrane. This uphill transport generates a proton concentration gradient across the inner membrane. Because of the outer membrane is freely permeable to protons, whereas the inner membrane is not, this pumping causes the pH of the mitochondrial matrix to become higher. Means the hydrogen ion concentration is lower than that of the cytosol and inner membrane space. And electrical potential across the inner membrane also results from the uphill pumping the protons outward from the matrix, which become negative with respect to the inner membrane space. Thus, free energy released during the oxidation of NADH or FADH2 is stored both as an electric potential and proton concentration gradient. Collectively, the proton motive force across the inner membrane, the movement of protons back across the inner membrane, driven by this force, is coupled to synthesis of ATP from ADP and inorganic phosphate by ATP synthase, which now we I am going to discuss in detail. The synthesis of ATP from ADP and inorganic phosphate driven by transfer of electrons from NADH or FADH2 to O2 is the major source of ATP in aerobic non-photosynthetic cells. Much evident shows that in mitochondria and bacteria, this process called oxidative phosphorylation depends on generation of a proton motive force across the inner membrane with electron transport proton pumping, the ATP formation occurring simultaneously. In the laboratory, for instance, addition of oxygen and oxidizable substrates such as pyruvate or succinate to isolate intact mitochondria result in a net synthesis of ATP if the inner mitochondrial membrane is intact. So, in the presence of minute amount of detergent that makes the membrane leaky, electron transport and oxidation of these metabolites by oxygen still occurs, but no ATP is made. Under these conditions, no transmembrane protons concentration gradient or membrane electrical potential can be maintained. Now, first we discuss the magnitude of proton motive force, then the component of the respiratory chain and pumping of proton across the inner membrane. So now in the next section I discuss the structure of the ATP synthase and how it uses the proton motive force to synthesize ATP. I also consider how mitochondrial oxidation of NADH and FADH2 is controlled to meet the cell's need for the ATP. In electron transport chain, mainly we discuss about three topics. The first is the proton motive force is mitochondria is due to largely to voltage gradient across the inner membrane. As we have seen that the proton motive force is the sum of a transmembrane proton concentration gradient and electrical potential or voltage gradient. The relative contribution of the two components of the total PMF depends on the permeability of the membrane to ions other than protons. A significant voltage gradient can develop only the membrane is poorly permeable to other cations and to anions, as in the inner mitochondrial brain. So in this case, the developing voltage gradient soon prevents further proton movement, so only a small pH gradient is generated. So in contrast, a significant pH gradient can develop only if the membrane is also permeable to a major anion such as chloride ion, or if the proton ion are exchanged for another cation such as potassium ion. In either cases, proton movement does not lead to the voltage gradient across the membrane because there is always an equal concentration of positive and negative ions on each side of the membrane. This is the situation in the chloroplast thylakoid membrane during photosynthesis, as we discussed any other day. Compared with the chloroplast, then greater portion of the PMF in mitochondria is due to the membrane electrical potential, and actual pH gradient is the smaller. Now we move to the Next is electron transport in mitochondria coupled to proton translocation. The coupling between electron transport from NADH or FADH2 to oxygen and proton transport across the inner mitochondrial membrane, which generates the proton motive force, also can be demonstrated experimentally with the isolated mitochondria. As soon as oxygen is added to the suspension of mitochondria, the medium outside the mitochondria becomes acidic during electron transport from NADH to O2. Protons translocate from matrix to the inner membrane space. Since the outer membrane is freely permeable to protons, the pH of the outside medium is lowered briefly, and the measured change in pH indicates that about 10 protons are transported out of the matrix for every electron pair transferred from NADH to oxygen. And next is the electrons flow from FADH2 and NADH2 to oxygen through a series of 4 multiple complexes. We now examine more closely the energetically favored movement of electrons from NADH and FADH2 to the final electron acceptor O2. The respiring mitochondria, each NADH molecule releases 2 electrons to the respiratory chain. These electrons ultimately reduce one oxygen atom, half of an oxygen molecule, forming one molecule of water. As electron moves from NADH to O2, their potential decline by 1.14 volt, which correspond to the 26.2 kilocalorie per mole of electrons transferred, or in general 53 kilocalorie per mole for a pair of electrons. As noted earlier, much of this energy is conserved in the proton motive force generated across the inner mitochondrial membrane. Each of four large multi-protein complex in the respiratory chain span in the inner mitochondrial membrane and contains several cristae groups that participate in moving electrons. These small non-peptide organic molecules or metal ions are tightly and specifically associated with the multi-protein complexes, and shown into the next slide. So I'm showing this actually. So here this table is showing the electron carrying cristae groups in the respiratory chain. So before considering the function of each complex, we examine several of these electron carriers. So this is the table where the protein components and their cristae groups are shown here. So complex 1 is NADH coenzyme Q reductase, complex 2 is succinate coenzyme Q reductase, complex 3 is coenzyme Q H2 cytochrome C reductase, and complex 4 is cytochrome C oxidase. And these are the protein groups for this. So now I am going to discuss one by one all these complexes. So first the complex 1. So electrons are carried from NADH to coenzyme Q by NADH coenzyme Q reductase complex. NAD plus is exclusively 2 electron carrier. It accepts or releases a pair of electrons at a time. In the NADH coenzyme Q reductase complex, electrons first flow from NADH to and flavin mononucleotide, a cofactor related to the FAD, then to an iron-sulfur cluster, and finally to the coenzyme Q. FMN, like FAD, can accept 2 electrons but does so 1 electron at a time. The overall reaction catalyzed by this complex is NADH plus coenzyme Q plus 2 protons is gives to the NAD plus plus proton and coenzyme Q H2 which is in reduced form. And now we come to the complex 2. Complex succinate dehydrogenase, the enzyme that oxidizes a molecule of succinate to fumarate in the citric acid cycle, is an integral component of the succinate coenzyme Q reductase complex. The two electrons released in conversion of succinate to fumarate are transferred first to the FAD, then to the iron-sulfur cluster, and finally to the coenzyme Q. The overall reaction catalyzed by this complex is succinate plus coenzyme Q gives the fumarate and coenzyme Q H2. Now we come to the complex 3. A coenzyme QH2 generated either by complex 1 or complex 2 donates 2 electrons to the coenzyme QH2 cytochrome C reductase complex, regenerating oxidized coenzyme Q. Concomitantly, it releases 2 protons picked up on the cytosolic phase into the inner membrane space, generating part of the proton motive force. Within a complex 3, the released electrons first are transferred to an iron-sulfur cluster within complex 3 and then to the B-type cytochrome or cytochrome C1. Finally, the 2 electrons are transferred to 2 molecules of the oxidized form of cytochrome C, a water-soluble peripheral protein that diffuse in the inner membrane space. For each pair of electron transferred, the overall reaction catalyzed by the coenzyme QH2 cytochrome C reductase complex is coenzyme QH2 plus 2 cytochrome C3. It gives to coenzyme Q plus 2 protons plus 2 cytochrome C3 plus. Now the last complex 4 is cytochrome C. After being reduced by the coenzyme QH2 cytochrome C reductase complex, the transports electrons one at a time to the cytochrome C oxidizing complex. Within this complex, electrons are transferred again one at a time, first to a pair of copper ion called Cu plus 2, then the cytochrome A, next to a complex of another copper ion Cub plus 2, and then cytochrome A3, and finally to the O2. The ultimate electron acceptor yielding H2O. For each pair of electrons transferred, the overall reaction catalyzed by the cytochrome C oxidase complex is 2 cytochrome C plus 2 plus 2 protons plus half O2. This gives you 2 cytochrome C plus 3 plus water. So, during the transport of each pair of electrons through the cytochrome C oxidase complex, 2 protons are translocated across the membrane. Here I am giving you the overview of multi-protein complex bound to prosteroid groups and associated mobile carriers in the respiratory chain. The blue arrow indicates the electron flow. The red arrow indicates the proton translocation. So, pathway from NADH, the left part. A total of 10 protons are translocated per pair of electrons that flows from NADH to oxygen, NADH only to H2O, to O2. The protons released into the matrix space during oxidation of NADH by NADH coenzyme Q reductase are consumed in the formation of water from O2 by cytochrome C oxidase, resulting in no net proton translocation from the reactor. Now we come to the right pathway from the succinate. During oxidation of succinate to fumarate and reduction of coenzyme Q by the succinate coenzyme Q reductase complex, no protons are translocated across the membrane. The remainder of the electron transport from coenzyme QH2 process proceeds by the same pathway as in the left diagram. Thus, for every pair of electrons transported from succinate to O2, 6 protons are translocated from the matrix into the inner membrane space. Coenzyme Q and cytochrome C function as mobile carriers in electron transport from both NADH and succinate. Now we comes to our ATP synthase. So the hypothesis that proton motive force across the inner mitochondrial membrane is the immediate source of energy for ATP synthesis was proposed in 1961 by Peter Michaeli. Virtually all researchers working in oxidative phosphorylation and photosynthesis initially opposed this hemosmotic mechanism. They favored a mechanism similar to the well-educated substrate-level phosphorylation in glycolysis, in which oxidation of a substrate molecule is directly coupled to ATP synthesis by analogy. Electron transport through members of chloroplast or mitochondria was believed to generate an intermediate containing a high energy chemical bond, that is phosphate linkage into an enzyme by an ester bond, which was then used to convert PI and ADP to ATP. Despite intense effort by a large number of investigators, however, no such intermediate was ever identified. So, definitely evidence supporting the role of proton motive force in ATP synthesis awaited development to techniques to purify and reconstitute organelle membrane and membrane proteins. The experiment with chloroplast thylakoid vesicles containing F0 F1 particle outlined the one of several demonstrating that F0 F1 complex in ATP generates enzyme, and that ATP generation is dependent on proton movement down an electrochemical gradient with general acceptance of Michaeli chemoosmotic mechanism. Researchers turn their attention to the structure and operation of the F0 F1 complex. Now we comes to its structure, that ATP synthase comprises two multi-protein complexes termed as F0 and F1. So the F0 F1 complex or ATP synthase has two principal components, F0 and F1, both of which are multimeric proteins, which is shown in here. The F0 component contains three types of integral membrane protein designed as A, B, and C. In bacteria and in yeast mitochondria, the most common subunit composition is A1, B2, C10. But F0 complex is an animal mitochondria have 12 C subunits and those in chloroplast have 40. In all cases, the C subunit form donut-shaped ring in the plane of the membrane. The A and 2B subunits are visibly linked to one another, but not the ring of the C subunit. The F1 portion is water-soluble complex of 5 distinct polypeptides with the composition of alpha 3, beta 3, gamma, delta, and eta. The lower part of the F1 gamma subunit is coiled that fits into the center of the C subunit ring of the F0 and appears rigidly attached to it. The F1 Beta subunit is rigidly attached to gamma and also forms rigid contacts with the several of C subunit of F0. The F1 alpha and beta subunit associated in the alternating order to form a hexamer alpha beta alpha beta alpha beta, or we can say alpha beta the whole pair, which rests atop of the single long gamma subunit. The F1 delta subunit is permanently linked to one of the F1 alpha subunit and also the B subunit of F0. Thus, the F0 A and B subunit and the delta subunit and alpha beta का whole thread hexamer of the F1 complex form rigid structure anchored into the membrane. The rod-like beta subunit form a Cellular that prevents the alpha B whole thread hexamer from moving while it rests on the gamma subunit. So this all picture is showing here. So I have to see whether I have completed the things or not. So I have completed this. So when ATP synthesis is embedded in the membrane, the F1 component forms a knob that protrudes from the cytosolic phase as demonstrated in this picture. The submitochondrial vesicles from F1 is removed by mechanical agitation cannot catalyze ATP synthesis. When F1 participate reassociate with these vesicles, they will again become fully active in ATP synthesis. Because F1 separated from the membrane is capable of catalyzing ATP hydrolysis, it has been called the F1 ATPase. However, its function in the cell is to synthesize ATP. So we examine how it does, how ATP is generated. So we will discuss into the next. So here the rotation of F1 gamma subunit driven by proton movement through F0 powers the ATP synthesis. Each of the three subunits in the complex F0 F1 complex can bind ADP and PI and catalyze ATP synthesis. However, the coupling between proton flow and ATP synthesis must be indirect, since the nucleotide binding site on the beta subunit of F1, where ATP synthesis occurs, are 9 to 10 nanometer from the surface of the mitochondrial membrane. The most widely accepted model for ATP synthesis by F0 F1 complex, the binding change mechanism is posited just such as indirect coupling. So this picture is the binding change mechanism of ATP synthesis from ADP and PI by the F0 F1 complex. This view is looking up at F1 from the membrane surface. Each of the F1 beta subunit alternate between three conformational states that differ in their binding affinity to ATP, ADP and PI. So in step 1, after ADP and PI bind to one of the three beta subunit whose nucleotide binding site is in the open, conformational proton flux powers a 120-degree rotation of the gamma subunit. This causes an increase in the binding affinity of beta 1 subunit for ADP and PI to L. This is the low, and an increase in the binding affinity of beta 3 subunit for ADP and PI from L to T tight, and a decrease in the binding affinity of the beta 2 subunit from ATP from T to O, causing release of the bound ATP. In step 2, the ADP and PI in the T site, here the beta 3 subunit from ATP, a reaction that does not require an input of energy, and ADP and PI bind to the beta 2 subunit, which is in the O state. This generates an F1 complex identical with the which started the process shown in the stream left, except that it rotates 120 degrees. Now, in step 3, another 120 degree rotation of gamma again causes the O to L to T to O conformational change in the beta subunit as described earlier. So, repetition of step 1 and 2 leads to the formation of 3 ATP molecules for every 360 rotation of gamma. So, up to here we have learned that how the ATP is generated, but from where this proton motive force is powered actually. So this is the thing. This is the chemoosmotic hypothesis. This picture here I'm showing you. So just a quick, not necessary, but you have to know this thing also. So in addition to powering ATP synthesis, the proton motive force across the inner mitochondrial membrane also powers the exchange of ATP formed by oxidative phosphorylation inside the mitochondria for ADP and PI in the cytosol. This exchange, which required for oxidative phosphorylation to continue, is mediated by two proteins in the inner membrane, a phosphate transporter and ATP-ADP antiporters. So as shown in this here is ATP-ADP antiporter and phosphate transporter. So this figure is the coordinate this figure shows the coordinate action of two antiporter is shown in purple and green, results in the uptake of one ADP and one HPO4 in exchange for one ATP, powered by the outward translocation of one proton during electron transport. The outer membrane is not shown here because it is permeable to molecules smaller than 5000 Dalton. The phosphate transporter catalyzes the import of one HPO4 coupled to the export of one hydroxyl ion. Likewise, the ATP-ADP antiporter allow one molecules of ADP to enter only if one molecules of ATP exists simultaneously. The ATP-ADP antiporter is a dimer of 230,000 Dalton subunit make up 10 to 15% of the protein in the inner membrane. So it is one of the more abundant mitochondrial protein. Functioning of the two antiporters together produces an influx of one ADP and one inorganic phosphate and influx of one ATP together with one hydroxyl ion. Each hydroxyl ion transported outward combines with a proton translocate during electron transport to inner membrane space to form water. This drives the overall reaction in the direction of ATP export and ADP and inorganic phosphate export. because some of protons translocated out of mitochondria during electron transport provide the power for ATP-ADP exchange. Fewer protons are available for ATP synthesis. It is estimated that for every 4 protons translocated out, 3 are used to synthesize one ATP molecule and 1 is used to power the export to ATP from the mitochondria in exchange for ADP and PI. This expenditure of energy from the proton concentration gradient to export ATP from the mitochondria in exchange for ADP and PI ensure a high ratio of ATP to ADP in the cytosol, where hydrolysis of high energy phosphoanhydride bond of ATP is utilized to power many energy requiring action. So here I am giving you last sentence for this whole video. The rate of mitochondrial oxidation actually not. It normally depends on ADP level. So from up to here, this I have finished this topic, energy transduction in mitochondria, and here is the key concept for this ATP synthesis concept. So I am not going to read this, and please you can read, and thanks for listening this informative video I have made. You all, thanks for listening also.